Dr. Bar and I love me some histology. We're going to talk about meninges and choroid plexus. By the end of the session, I want you to be able to describe the structure and function of the meninges and choroid plexus. The meninges are comprised of connective tissue membranes, and they cover the spinal cord and the brain. So from out to in, I'm going to show you here the dura mater, which is a very thick membrane that's covering the spinal cord and the roots here. You could also see it histologically as this dense pink connective tissue right here. The arachnoid mater is a very thin membrane that's right underneath the dura mater. There's a space between the two meninges, which is called the subdural space. Here you see the arachnoid mater, here's the subdural space, and underneath the arachnoid mater, you actually have your subarachnoid space filled here with cerebrospinal fluid. The pia mater is, again, a membrane that's closest to the white matter, and it's a very thin connective tissue, but also joined in with several blood vessels. In the brain, going from out to in, we have our dura mater, but you see here that there are two layers of the dura mater. There's a periosteal layer, which is closest to the bone, and then you have a meningeal layer, which actually curves around and covers the surface of the brain. You see here that you have this formation of the superior sagittal sinus between the two layers of the dura mater. Underneath the dura mater, you also have your arachnoid mater, which is also filled with cerebrospinal fluid and containing blood vessels as well. You can see that there are structures that actually are produced from the subarachnoid region here that protrude through the dura mater and into the sagittal sinuses, and these are called arachnoid granulations, and their role is to absorb any cerebrospinal fluid within the sagittal sinus here. The pia mater is a very thin membrane that's coating the surface of the brain here in the gray matter of the brain, and it's comprised of a thin pia mater along with the astrocyte foot processes right here. The choroid plexus is comprised of ependymal cells, which are ciliated cuboidal type cells. There's a thin layer of pia mater and fenestrated capillaries. The arrangements of the choroid plexus is the structures have a folding or projection type of arrangement, and their main function is to remove water and ions from the bloodstream and movement of that into the cerebrospinal fluid. That's why it needs to be fenestrated capillaries, which allow for adequate movement and passage of water and ions through and between the endothelial cells. Choroid plexus is located in various regions, the third and the fourth ventricles, particularly the roof area of these ventricles, as well as parts of the lateral ventricles. Recall that the ependymal cells have cilia, so the role of the cilia is to move the cerebrospinal fluid to the various compartments in the various ventricles. Here you see the histology of the choroid plexus with the projections. As you see here on lower power view, on higher power view, you see the layer of the ependymal cells. You have that pink, pale connective tissue, which is your pia mater, and then you have your capillaries centrally there. Some clinical correlations. Well, why do we need to know about choroid plexus and meninges? Remember that choroid plexus, the ependymal cells make our cerebrospinal fluid. So if there's overproduction of CSF, they can cause hydrocephalus, enlargement of the ventricles, and flattening of the adjacent brain. There can be other causes of hydrocephalus as well, such as tumors, which are, again, irrelevant to the choroid plexus pathology. With meningitis, the meninges can become infected by bacterial viral agents, causing inflammation of the meninges and influx of white blood cells such as neutrophils and lymphocytes. There can be also bleeding into the subarachnoid space via rupture of Berry aneurysms. You may have heard, in anatomy where they talk about rupture of Berry aneurysms can cause a very severe splitting headache. The meninges are very sensitive to chemical agents, so blood will cause it to become irritated. The inflammation will also cause irritation, and one common clinical presentation of meningitis and subarachnoid bleeding is not only headaches, but also nuchal rigidity or neck stiffness. Another clinical correlation is we want to access clinically the subarachnoid space. Number one, to possibly obtain cerebrospinal fluid for analysis, so we can perform a lumbar puncture. We also can administer medications such as anesthesia or chemotherapy. So again, in the lumbar puncture or administration of medications, we want to puncture through the dura mater to enter into the subarachnoid space. Here we're administrating medication, but we could draw also the cerebrospinal fluid out through the needle for our lumbar puncture and CSF analysis. So on that note, I will leave you with a recap of the mater, the clinical correlations, and the choroid plexus with its corresponding clinical correlation as well. Email me or contact me if you have any questions or need clarifications, and then I will see you at the next lecture. Thank you. Bye.